Number 1. Enumerate the modes of gene action and determine the phenotype and genotype of the progenies produced in each mode. So, complete dominant gene action. Complete dominant gene action occurs when the dominant allele is so strong that it produces its phenotype regardless of the genotype. Only a single dominant allele is needed to produce the dominant phenotype. This means the homozygous dominant and heterozygous genotypes both produce the dominant phenotype. Thus, the phenotypes produced by these genotypes are identical. The recessive allele can produce two recessive phenotypes only when no dominant allele is present, which means it can produce the recessive phenotype only when a fish is homozygous recessive. Next is incomplete dominant gene action. Incomplete dominant gene action occurs when the dominant allele expresses itself more strongly than the recessive allele, but it is not strong enough to completely suppress the recessive allele in the heterozygous genotype. Because of this, the dominant phenotype can be produced only when a fish has two copies of the dominant allele. Since the recessive allele is not completely suppressed by the dominant allele, the heterozygous genotype produces a phenotype that resembles, but it is not identical to the recessive phenotype. Is produced only when a fish is homozygous recessive because the heterozygous genotype produces a phenotype that is similar to but is different from dominant phenotype when the mode of gene action is incomplete dominance. Next is additive gene action. With only one exception, all qualitative phenotypes controlled by a single autosomal gene that have been discovered in cultured food fish are controlled either by complete or by incomplete dominant. The exception is a gene that controls golden palomino and normal body colors by additive gene action in rainbow trout. When the phenotypes are controlled by additive gene action, there is no dominant or recessive allele. Both alleles contribute equally to the production of the phenotype, so the heterozygous genotypes produce a phenotype that is intermediate between those produced by two homozygous genotypes. Consequently, when the mode of inheritance is additive gene action, there are three genotypes and three phenotypes, unique phenotype for each genotype. Number 2. Differentiate dominant from a recessive gene. Dominant is always expressed when present. Recessive is only expressed when no dominant genes are present. Individual receives two versions of each genes, known as alleles, from each parent. If the alleles of a gene are different, one allele will express it is the dominant gene. The effect of the other allele called recessive is masked. A dominant G allele in common car produce normal pigmentation, and the recessive G allele produce gold pigmentation. A cross between normally pigmented and gold common car produce all normal progenies. Letter A. What are the genotypes of the parents? The parents' genotype would be used as the basis for predicting the genotype of their offspring, which in turn may be crossed filial generation. What are the genotypes of the progenies? Letter C. What genotype should be mated to produce an odd gold phenotype? So it is the GG genotype to produce odd gold phenotypes. Based on the theory discussed in the hybrid cross, letter A. List all the gametes produced by the hybrid cross of two homozygous Greigapes with curves. Gametes are 4GCU. Letter B. Draw a Punnett square and determine the genotypic and phenotypic ratios are expected from the cross. A transparent female guppies can be produced if the male is transparent and the female has at least XEH allele, as the transparent male guppies are produced only when female has the recessive XEH allele. Number 6. Determine the factors that cause variation among organisms. Mutations, the changes in the sequence of genes in DNA, are one source of genetic variations. Another source is gene flow, or the movement of genes between different groups of organisms. Finally, genetic variation can be a result of asexual reproduction, which leads to the creation of new combination of genes. 7. Understand and apply the concept selection in fish stock management. 8. What is genetic drift and how can it be prevented? Genetic drift is a random change in allele frequencies with the population. It is one of the main mechanisms that drives evolutions. Genetic drift can be prevented by the regular replacement of foundations animals from genetically fixed embryos in the only way to effectively slow down genetic drift and prevent genetic changes from the accumulating.